Chapter number 10, Cell Cycle and Cell Division. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell that goes on to form such a large organisms. Growth and reproduction are characteristics of cell, indeed, of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. The newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cell cycles of growth and division allow a single cell to form a structure consisting of a millions of cells. Cell cycle. Cell division is very important process in all living organisms. During the division of a cell, DNA replication and cell growth also take place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cell containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells is termed cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesizes other occurs only during one specific stage in the cell cycle. The replicated chromosomes, DNA, are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. 10.1.1 Phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. However, this duration of cell cycle can vary from organisms to organisms and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases, interphase and second is M phase, that is called mitosis phase. The M phase represents the phase when the actual cell division or mitosis occur, and the interphase represents the phase between two successive M phases. It is significant to note that in the 24-hour average duration of cell cycle of a human cell, cell division proper lasts for about an hour. The interphase lasts more than 95% of the duration of cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, karyokinesis, and usually ends with the division of cytoplasm, that is cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases. First, G1 phase, second, S phase, and third, G2 phase. G1 phase corresponds to the interval between the mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows, but does not replicate its DNA. S or synthesis phase marks the period during which the DNA synthesizes or replication take place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell has diploid or 2n number of chromosomes at G1, even after S phase, the number of chromosome remains the same, that is 2n. In animal cells during the S phase, DNA replications begin in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Some cells in the adult animals do not appear to exhibit division, example heart cell, and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not further exist G1 phase to enter an inactive stage called quiescent stage G0 of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so, depending on the requirement of the organisms. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this, where haploid cells divide by mitosis. For example, male honeybee, against this, the plants can show mitotic division in both haploid and diploid cells. From your recollection of examples of alternation of generation in chapter number 3, identify plant species and stages at which mitosis is seen in the haploid cells. 10.2 M phase, that is called mitotic phase. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cells is the same, it is also called equational division. Though, for the convenience, mitosis has always been divided into 4 stages of nuclear division, that is called karyokinesis. It is very essential to understand that cell division is progressive process and very clear cut lines cannot be drawn between various stages. Karyokinesis involves following 4 stages. First is prophase, second is metaphase, third is anaphase, and fourth is telophase. 10.2.1 Prophase. Prophase, which is the first stage of karyokinesis of mitosis, follows the SNG2 phase of interphase. In the SNG2 phase, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centrosome, which had undergone duplication during the S phase of interphase, now begins to move towards opposite poles of the cell. The completion of prophase can thus be marked by the following characteristic events. First, chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of 2 chromatids attached together at the centromere. Second, centrosome, which had undergone duplication during interphase, begins to move towards opposite poles of the cell. Each centrosome radiates out microtubules called asters. The 2 asters together with spindle fibers form mitotic apparatus. Cells at the end of prophase, when viewed under the microscope, do not show Golgi apparatus, endoplasmic reticulum, nucleolus, and nuclear envelope. 10.2.2 Metaphase. The complete disintegration of the nuclear envelope marks the start of second phase of mitosis. Hence, the chromosomes are spread throughout the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and they can be observed clearly under the microscope. This then is the stage at which morphology of chromosome is most easily studied. At this stage, metaphase chromosome is made up of 2 sister chromatids, which are held together by centromere. Small disc-shaped structures at the surface of the centromere are called kinetochores. These structures serve as the site of attachment of spindle fibers, formed by the spindle fibers to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator with 1 chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole and its sister chromatids connected by its kinetochore to spindle fibers from the opposite pole. The plane of alignment of the chromosomes at metaphase is referred to as metaphase plate. The key features of metaphase are: first, spindle fibers attached to kinetochores of chromosomes. Second, chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fibers to both poles. 10.2.3 Anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the 2 daughter chromatids, now referred to as daughter chromosomes of the future daughter nuclei, begin their migration towards the 2 opposite poles. As each chromosomes move away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and hence at the leading edge, with the arms of chromosome trailing behind. Thus, anaphase stage is characterized by the following key events. First, centromere split and chromatids separate. Second, chromatids move to opposite poles. 10.2.4 Telophase. At the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosomes can no longer be seen, and each set of chromatin material tends to collect at each of the 2 poles. This is the stage which shows the chromosomes following key events. First, chromosomes cluster at opposite spindle poles and their identity is lost as discrete elements. Second, nuclear envelopes develops around the chromosome cluster at each pole forming two daughter nuclei. Third, nucleolus, Golgi complex and ER reform. 10.2.5 Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, that is karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which the cell division gets completed. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center cell, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall, therefore, they undergo cytokinesis by a different mechanism. In plant cell, wall formation starts in the center of the cell and grows outwards to meet the existing lateral walls. The formation of the new cell wall begins with the formation of simple precursor called the cell plate that represent the middle lamellae between the walls of two adjacent cells. At the time of cytoplasmic division, organisms like mitochondria and plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by the cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium. Example, liquid endosperm in a coconut. 10.3 Significance of mitosis. Mitosis or the equational division is usually restricted to the diploid cells only. However, in some lower plants and in some social insects, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some examples where you have studied about haploid and diploid insects? Mitosis usually results in the production of diploid daughter cells with identical genetic complement. The growth of multicellular organisms is due to the mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It is therefore becomes essential for the cells to divide to restore the nucleocytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cells of the upper layer of the epidermis, cells of the lining of the gut, the blood cells are being constantly replaced. The mitotic divisions in the meristematic tissue, the apical and the lateral cambium, results in the continuous growth of the plant throughout their life. 10.4 Meiosis. The production of offspring by sexual reproduction includes the fusion of the two gametes, each with the complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. The specialized kind of cell division that results reduces the chromosome number by half, results in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key feature of meiosis are as follows. First, meiosis involves two sequential cycles of nuclear end cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. Second, meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Third, meiosis involves pairing of homologous chromosome and recombination between the non-sister chromatids of homologous chromosome. And the last one, four haploid cells are formed at the end of meiosis 2. 10.4.1 Meiosis 1. Prophase 1. Prophase of the first meiotic division is typically longer and more complex when compared to the prophase of mitosis. It has been further subdivided into the following 5 phases based on the chromosomal behavior, that is liptotin, zygotin, phagotin, diplotin, and diakinesis. During liptotin stage, the chromosome becomes gradually visible under the light microscope. The compaction of chromosome continues throughout the liptotin. This is followed by the second stage of prophase 1 called zygotin. During this stage, chromosomes start pairing together and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate that chromosome synapsis is accompanied by the formation of complex structure called synaptonemal complex. The complex formed by a pair of synapsed homologous chromosome is called a bivalent or a tetrad. However, these are more clearly visible at the next stage. The first two stages of the prophase 1 are relatively short lived compared to the next stage, that is phagotin. During this stage, the four chromatids of each bivalent chromosomes become distinct and clearly appears as tetrads. This stage is characterized by the appearance of recombination nodules, the sites at which crossing overs occurs between the known sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme mediated process, and the enzyme involved is called recombinase. Crossing over leads to the recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of phagotin, leaving the chromosome linked at the sites of crossing overs. The beginning of diplotin is recognized by dissolution of synaptonemal complex and the tendency of the recombined homologous chromosomes of the bivalents to separate from each other except at the site of crossovers. These egg-shaped structures are called chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. The final stage of meiotic prophase 1 is dikinesis. This is marked by terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of dikinesis, the nucleolus disappears and the nuclear envelope also breaks down. Dikinesis represents transition to metaphase. Metaphase 1. The bivalent chromosomes align on the equatorial plate. The microtubules from the opposite poles of the spindle attach to the kinetochore of homologous chromosomes. Anaphase 1. The homologous chromosomes separate while sister chromatids remain associated at their centromeres. Telophase 1. The nuclear membrane and the nucleolus reappear, cytokinesis followed, and this is called dyad of cell. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended stage of interphase nucleus. The stage between the two meiotic division is called interkinesis and is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by prophase 2, a much simpler prophase than prophase 1. 10.4.2 Meiosis 1. Prophase 2. Meiosis 2 is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to meiosis 1, meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 2. The chromosomes again become compact. Metaphase 2. At this stage, the chromosomes align at the equator and the microtubules from the opposite poles of the spindle gets attached to kinetochore of sister chromatids. Anaphase 2. It begins with the simultaneous splitting of centromere of the centrosome, which was holding the sister chromatids together, allowing them to move towards opposite poles of the cell by shortening of microtubules attached to the kinetochore. Telophase 2. Meiosis ends with the telophase 2, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is, 4 haploid daughter cells. 10.5 Significance of meiosis. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across generations in sexually reproducing organisms, even though the process per se paradoxically results in the reduction of chromosomes number by half. It also increases the genetic variability in the population of organisms from one generation to the next. Variations are very important for the process of evolution. Thank you.